Hello guys, my name is Dr. Varun and here I am back with another video for 30 days, 30 most important topic for NEET 2025 Biology.
Chalo, let's complete it.
Well, in today's video, we're gonna be learning about how the nerve impulse is being generated and conducted over a neuron. Well, for this topic, we have to know certain pointers that during the normal time, the neurons are their resting state. Means, for example, if I about to pinch you. So when I will pinch you, what will happen? There is an impulse goes that there is someone is pinching, that sir is pinching. So what you will do? You will do your hand like this. But if your friend would be doing, there would be a slap on his face. But here we come back to the major question. That is what? That in the major question is that once, if there is a neuron present all over our body, okay, nerve net, okay, how when we touched it and it caused the feeling of sensation that has been taken up to the brain and accordingly the functions comes back, okay. But from here to brain, multiple neurons are there. So how within a neuron is also a bigger cell? So we don't know which part of the neuron is being stimulated, how it is stimulated, and how the nerve impulse will conduct. In one neuron we are seeing, and in the next one, that is what? Chemical synapses or the electrical synapses, which is not our topic for today in detail, but at least within the neuron, how the impulse is generating, that is our topic. Let's just come back to the topic. So during the time when the pinch has not been started, your neurons of your hands or any organ which has been excited, it is what? It is their resting state. So the neurons are during the resting state. So first we are knowing about this. Now during this time, what basically happened? Your neuron is doing nothing. But during nothing also there is certain amount of potential present. Potential means what? Potential means a electrical difference that is being produced over there. If you have learned the physics, and I know that you might not be learning the physics, but you should know about the physics, that resting potential means the difference between the charge. Okay, that is a potential. So here what happened? Imagine that this is your axon. So axon must be like this, what? Your neuron, of course. So there will be what? Big axon. There is like, imagine that this is what? This, my hand is the cell body. This is the axon, and here there will be what? Cell endings. Okay. So I am what? I am concerned with this structure. Of course, here also it's a similar thing will happen, but in our book, this area is being shown. So we will show it like this. So if this is the axon, okay, axon part of it. So outer a membrane is present, we call it axon lemma. Inside cytoplasm present, would we call it exoplasm? Then where this potential coming, sir? Major question. So the potential is present on this membrane. So the membrane outside area has what? A positive charge. Inside have a negative charge. How this charge is being done? Because of the charge difference. Means outside there is extra positive charge, inside there is a negative charge. Who is doing it? Ions are doing it in our body. So we are making what? I am making a neuron. Okay, over here. Just see. Always remember, the cell has what? Certain receptors present. Okay. So this is, imagine this is your neuron. Okay. On this neuron, I said what? There are certain receptors present. Means you know that if the lemma, plasma membrane is there, means there will be a certain proteins present which act like a channel so things can go in and out. So what are those things present? So there is this one channel present, other channels, this is one other type of channel present, this is another type of channel present, certain channels which are always open, they are also present at regular intervals. So I have made it like this. Okay, here of course space is less now, so I am making like this. So these are the different channels present. Let's just name it accordingly. So this is the white channel is the diffusion channels. So diffusion channels, what will happen? Sodium, potassium going in, going out, no problem. Okay, what happened other time? This yellow color channel that you are seeing, it is called as sodium potassium pump. It requires ATP for its working. Okay, so this sodium potassium pumps do what? It throws out. So this sodium potassium pumps throws out 3 sodium outside from the inside of the axon area, whereas simultaneously it is taking inside 2K positive. Now tell me, 3 positive going out, 2 positive coming in means what? Outside more positive, inside less. That gives what? The outside region. I told you na, this, imagine that this is the axon lemma. So outside area will be positive, inside area would be what? Negative. After this there won't be any charge. Okay, it will be diffused, neutralized by the positive negative of the tissue. Okay, just on the membrane and closer to it area would be what? Positive on the outside, inside it is negative. This area is what? No charge, diffused. Okay, so this becomes what? Inside more. So I am just enlarging it, and inside becomes what? Inside of the axon becomes negatively charged. Of course it is positive charge only, but the difference is there. Okay, so difference causes it less positive. So inside membrane is less positive, whereas outside membrane is what? Positively charged. By this we have to remember certain thing, that the membrane, when it is on resting phase, it is permeable, because we know that membranes are permeable. Permeable means it allow the things to come inside or go out, right? So when it is at the resting phase, this membrane, mean axon lemma membrane, is impermeable to negatively charged proteins which are present inside the neuron, because it's a cell will have proteins. Negatively charged proteins means negatively charged proteins are going nowhere, not allowed, whereas it is permeable to K positive ions to come inside, come, because it is coming also inside, where it is nearly impermeable to sodium ions. Normally it is going out with the help of pump. So this thing you have to remember. So resting phase is about this. Moving on to the next thing. What is the next thing? What will happen when there is excitation? Means when the stimulus comes, means pinching start. So this pinching thing is called as what? It is called as excitable state or phase. During the excitable phase, this neuron will change. Now think yourself, if I will pinch for a certain period of time, if you are writing your and I pinch, you will like everything changed, and you will like without seeing even that it's your sir. Okay, so this thing happens. Same thing if someone just give a slap for a moment of few seconds, milliseconds, what happened? You'll be like, oh, okay, what is happening? Okay, like this happens now. So same thing with the over here in the excitable phase. If you give a stimulus, this will change. Normally it is doing what? Sodium outside, K positive inside. Here what happened? This stops. Whatever it is doing stop, and the sodium gates open up, which is another gates. So these gates are sodium voltage gated channels, which will open up. Sodium start going inside. Sodium started going inside. This is just opposite. Yes, my friend, this is just opposite, that sodium started coming inside the neuron, and as it is started coming inside in that area which is being excited, that side reversal of polarity will take place. Means more positive coming inside, outside nothing going. Even K positive is also not going outside. Sodium pump is also stopped in the starting, I have told you. So what will happen? Inside area will become more positive. Reversal of polarity takes place over here. So this site is called as, it becomes depolarized. Polarization changed. Okay, so let's just see that when the excitation happens. So over here I am making again a neuron for you. So this is the neuron. Let's just make it a little larger. So this is your axon. I'm just for the representation purpose. So what was happening? You remember that here is, we have, this is sodium potassium pump. Then we have this thing. And this gate, I have shown you 3 types of gates. Okay, and the diffusion gates are everywhere. Okay. Now, in this state, the first point that I told you was what? It was that these gates, that is sodium potassium pump, will close. If it is closed, means nothing going inside, nothing going outside. For example, if the stimulus coming over here. So this area is what? Where the stimulus come. Okay, and let's show the stimulus with certain yes, like this. Okay, this is the area where the stimulus come. Okay, now if the stimulus came over here and this stops, and I told you what will happen, sodium voltage gates will open up, and this pink color is what? Sodium voltage gated channel opens up. As they will open up, what will happen, beta? The sodium, rapid influx of sodium will occur. So sodium influx will occur. And as the sodium went inside, what will happen in this area? It changed the polarity. Now inside become what? Positively charged, this whole area. Whereas outside over here will become what? Negatively charged. But over there, inside was always negative. Next area would be still negative. Outside would be what? Positive. It will still same. Okay. So at this state, as there is a reversal of polarity, so reversal of polarity occur. And we call this area, this site is depolarized. I hope you understand this point. And this area now would be what? Action potential that is being generated. And this area, what will happen? The positive charge will move towards the next negative. This will come back to this side. And this is how the impulse will keep on generating till the end. So this is how it will conduct. Conduction will take place. So from here we understood what? That once the stimulus come, reversible of polarity occur because all the other gates stops and sodium voltage gated channel open up, sodium coming inside, positive inside, negative outside, reversibility. This area is called as depolarized. Now this area, since inside the depolarized, it will stay. This sodium channels will only open for 0.5 milliseconds. Sodium voltage gated channels only opens for 0.5 seconds. Now it will close, and now since inside it what is there? Positive charge, outside is negative. Now the thing is that, do you want to be always in excited state? Someday is dull or you will come back to the normal. If I crack a very PJ, you will be like, but if I crack a really good joke, some kind of a sarcasm, you will be like, dude, that was like, ha ha ha, something like that, right? Same thing over here. Okay, so it will come back to the, again, within the few point of a time. Even I give a what kind of joke you want, you can think of. Okay, so this area will come back to the normal, and that is called as repolarization. Now for repolarization happen, what happened? This blue color gates open up. Okay, blue color gates will open up. Okay, I'm just making a rough one this time. Okay, not exactly every gates. All the other gates would be closed. So what will happen? This potassium voltage gated channel opens up now. As they will open up, what will happen? From inside, the K positive, we know that inside K positive is high because when the resting phase, K positive coming inside, sodium going out. So what will happen? The sodium won't go from out, the K positive will go out. Will remove out. Since it is a positive charge, when the positive charge will go out, what will happen? Is outside, which was here at this time, you know na, inside it was positive, more positive, outside it was negative. So what happened? This keep on going out. As it will keep on going out, what will happen? Outside becomes more positive, inside remain what? Nothing is coming inside, so negative. So here what will happen? After certain time, K positive means positive charge outside become more and inside less, making polarity back to resting phase. Now if these channels keep on opening for the longer duration, what will happen? It will go more negative inside, and we call it hyperpolarization, which is not in your syllabus. But what is there in syllabus? Then this is what I'm gonna write it. That is what is the resting phase potential? The potential is minus 70 millivolt. What we are writing? What is the potential on the inside? Okay, then excitable phase. During the excitable phase, here, over here, the change that has occurred, what is the potential over here? It can goes to plus 35 millivolt. That is the action potential. First it will of course goes to 0, then it goes plus, okay, and the repolarization will come back to minus 70 millivolt over that area. And if it is hyperpolarization, then it goes further negative and it can go up to minus 90 millivolt. That's it. I hope you like this video. If you're able to understand something, share this with your friends if you really want to. Well, that's all for this video. Give it a subscribe if haven't yet. Give me a like, comment down if you're able to understand, or if you're not able to understand, then also there. I try to sum it up as easy as possible with less words and less information just required for NCERT. Well, that's all for this video. I taken over. Thank you.